Can we all rise as the academic procession files in? We're going to take the anthems, starting with the anthem of the Federal Republic of Nigeria, followed by the University of Ibadan anthem.
Nigeria, we hail thee, our own dear native land. Though tribe and tongue may differ, in brotherhood we stand. Nigerians old and proud to serve our sovereign motherland.
It's on that note I would like to invite the Dean of Faculty of Renewable Natural Resources to kindly recite the biodata of our lecturer today, while I request that he should rise as his biodata has been recited. Thank you.
Citation of Dr. Alfred Osai Onefeli. Dr. Alfred Osai Onefeli was born on the 24th of May, 1983, to late Mr. Francis Onefeli and Mrs. Lydia Onefeli of Amai Umubu in Ukwani local government area of Delta State, Nigeria. He completed his primary education at St. Peter Anglican Primary School, Oloruntele, Ileoluji, Okegbo local government area of Ondo State in 1995, and his secondary school education at Community High School, Oloruntele Ileoluji Okeigbo local government in Ondo State in 2001. Dr. Nefeli was admitted into University of Ibadan in 2004 to study forest resources management and completed his studies in 2009. He had his National Youth Service in Federal College of Agriculture, Ishiago, Ebonyi State in 2010-2011, where he won the Governor's Award as the best serving copper in the state. I hope we'll put our hands together for him. Dr. Nefeli obtained his MSc degree in Forest Biology and Silviculture in 2012 and his PhD in Forest Taxonomy and Agroforestry in 2021, all from the University of Ibadan. So he's actually a son of the soil. He had his first appointment with Edmond Nigeria Limited in Warri Delta State in 2012 as a forest officer and plant identifier. And he joined the University of Ibadan in 2015 as an assistant lecturer in the then Department of Forest Resources Management. He was promoted to the grade of Lecturer 2 in October 2018, Lecturer 1 in 2021, and Senior Lecturer in 2024. During his PhD, Dr. Nefeli was awarded a TETFund grant under the Academic Staff Training and Development Intervention Scheme of the University of Ibadan, which enabled him to undertake the taxonomy component of his research at Makerere University, Uganda, between 2017 and 2018. And he was nominated by the then Dean of the Faculty, Professor Bio Omitoyin, to participate in the Erasmus program at Vitatus Magnus University Agriculture Academy in Lithuania, where he studied molecular taxonomy from 2018 to 2019. In 2024, he won a postdoctoral fellowship as field team lead and postdoctoral scholar in the Conservation, Ecology, and Biogeography Lab, Department of Ecology, Montana State University, Bozeman, Montana, USA. The fellowship supports his ongoing work on taxonomy and a comprehensive floristic survey of the Cross River National Park, Afi River Wildlife Sanctuary, and the Mbe Mountains Community Forest in southern Nigeria. Dr. Nefeli served as a member of the undergraduate program committee in the Fund Department of Forest Resources Management, now Department of Forest Production and Products, and then Department of Social and Environmental Forestry from 2015 to 2017. He was a member of the congregation between 2016 and 2017, a member of the Students' Welfare Committee, Faculty of Renewable Natural Resources, the coordinator on the Graduate Studies Program for his department from 2019 till date, member of the Faculty Workshop Committee from 2020 till date, a member of the Departmental Welfare Committee, Departmental Postgraduate Committee, Departmental Information and Communication Technology Committee, all from 2021 till date. In 2023, he was appointed as the Assistant All Warden of Ransom Kuti All, a position he still holds. So the Kutites will be very happy to have their All Warden. Dr. Nefeli was the Faculty of Renewable Natural Resources representative of the Academic Staff Union of University, UI branch, from 2021 to February 2025. And in 2021, he became the staff advisor to Tree Club Association, University of Ibadan. In 2023, he served as collation officer for INEC during the general election, and is also the staff advisor to guidance of the nation's International Leadership Club, University of Ibadan, a role he continues to hold. He served as a member of the editorial board of the Second International Society of Tropical Foresters Conference in 2024, and is appointed as Departmental Wellness Officer. Dr. Nefeli is a member of several professional associations, including a live membership of the Forestry Association of Nigeria, a member of Society for Conservation Biology, Commonwealth Forestry Association, International Union of Forestry Research Organization, and International Society for Tropical Foresters. He has supervised over 23 BSc projects, 6 MSc dissertations, and is currently supervising a PhD, 4 MSc, and 7 BSc students. Can we put our hands together for him? Dr. Onefeli's personal hobbies include playing the keyboard, so he's a musician, he sings, he likes to counsel. You can see why he's a good undergraduate coordinator, and he's also into photography. Dr. Onefeli is happily married to Mrs. Oluchukwu Joy Onefeli, and the marriage is blessed with a promising daughter of Zion, Esther Ifoma Onefeli. So it's my pleasure to present to you this afternoon Dr. Alfred Osai Onefeli for his presentation.
The Dean, Faculty of Renewable Natural Resources, the Head of Department of Forest Production and Products, heads of other departments, the faculty officer, distinguished ladies and gentlemen. It is a great privilege for me to stand before this great audience today, and I do not take it for granted. To God be the glory, honor, and adoration. I thank the Dean, Faculty of Renewable Natural Resources, Professor Adejoke Olukemi Akinyele, and the Head of Department of Forest Production and Products, Professor A.O. Omole, for nominating me to present the 2024-2025 edition of the faculty lecture. This lecture, titled Where Taxonomy Meets Silviculture, Pathways to Sustainable Forestry in Nigeria, is the fifth from the Faculty of Renewable Natural Resources and the second from the Department of Forest Production and Products. The first faculty lecture from the department was presented by the current Dean of the faculty, Professor Adejoke O. Akinyele. A lecture was titled Achieving Sustainable Development Through Silviculture, Focus on Tree Domestication. My journey into the field of forestry and taxonomy began long before I entered the university. As a young student at Community High School, Olorun-Tele, in Ilelujo-Okegbo local government area of Ondo State, I found myself fascinated by how my agricultural science teacher pronounced the botanical names of trees during our lessons. The rhythm and elegance of those Latin names caught my attention, and I quietly began to dream of one day working in a field where such knowledge is regularly used. This interest aligned perfectly with my late father's vision. My father, Mr. Francis Onefeli of blessed memory, owned a kola nut farm near a forest reserve. He had always wished that one of his children would study a course that would help protect that forest and to stop illegal tree felling. Unfortunately, he could not afford to send me to university, but fate smiled on me. After finishing my secondary education, my eldest brother, Peter Onefeli, bought a JAMB form on my behalf and promised to sponsor my university education. When he asked me what course I would like to study and at which university, without hesitation, I chose forestry as both my first and second choices, selecting the University of Ibadan, Federal University of Technology, Akure, respectively. By divine arrangement, I was working on the field with my father one day when a friend, already a student of agriculture at the University of Ibadan, brought a copy of Punch newspaper. To my joy, my name was among the 4 merit students admitted to study forest resources management at the University of Ibadan. That moment was one of joy and fulfillment for me and my father, and also marked the beginning of my forestry career. After my bachelor's degree, I enrolled for a master's in forest biology and silviculture under the supervision of our current dean, Professor Adejoke Akinyele. While I enjoy silviculture, I still had a deep desire to work with the scientific names and classification of forest plants. Initially, I considered registering for my PhD in the Department of Botany to focus purely on plant taxonomy, but due to uncertainty and advice received, I stayed within forestry. Still, I was determined to find a way to blend taxonomy with silviculture. I approached Professor G.A. Lamid of the Department of Wildlife and Ecotourism to introduce me to a taxonomist who could possibly co-supervise my thesis. I met Professor A.E. Ayodele from the Department of Botany, and we had meaningful discussions. But since I had not carried my main supervisor along in the plan, it didn't work out. I dropped the idea of former co-supervisor but didn't drop my passion. To strengthen my taxonomic knowledge, I audited all the undergraduate and postgraduate courses in taxonomy at the Department of Botany, and I collected research materials from Professor Ayodele. Later, I contacted Professor J.D. Olowokudejo at the University of Lagos, who had supervised Professor Ayodele's PhD. I shared my background and interest with him, and he invited me to Unilag. He generously gave me many of his publications and encouraged me. I therefore continued my PhD research under the diligent supervision of the Dean of the Faculty of Renewable Natural Resources. With her guidance, I was able to integrate my passion for taxonomy into forestry-focused research. At the final defense, it was especially fulfilling to have Professor A.E. Ayodele serve as one of the examiners given his background in taxonomy. His recognition of some of the taxonomic insights in my work added further value to what had already been a well-supported academic journey. That experience strengthened my resolve to promote this integration of taxonomy into forestry education and practice. While taxonomy has long been a part of forestry, it was not widely emphasized during my time at the University of Ibadan, largely due to staff shortages. This lecture builds on the conviction by exploring how integration of taxonomy and silviculture can provide sustainable solutions to Nigerian forest challenges. Now, what is taxonomy? While teaching the course FPP 324, Forest Taxonomy, I often tell my students that they must be able to name at least 30 plant species before they can pass the course. However, I once had a particular group of students who misunderstood the purpose of the course. They thought forest taxonomy was simply about memorizing the names of trees. One day, while browsing through my social media page, I came across a video made by some of these students. I believe some of them may be in this hall today. In the video, two of the students said, "We are forestry students. Of course, people expect us to know the names of all trees they play." What they were really expressing was, how am I expected to know and keep the names of all trees in my brain, as if I do not have other things to do? In response to such categories of students, I always explain that taxonomy is not just about cramming scientific names. There are various ways to tree identifications and other life forms that do not require memorizing names. In fact, plant identification is not limited to taxonomists alone. Field workers such as loggers, who usually work closely with forest trees, can equally identify many tree species, although they may lack the formal taxonomic training needed to identify unfamiliar plants. This is where taxonomists play a key role. One of their key tools is the taxonomy key, a major instrument used in plant identification. For instance, Onefeli and Oladele Akin in 2020 developed a taxonomy key based on leaf epidermal structures to identify some species of Steculia in Nigeria, Table 1. Since taxonomy also involves the naming of plants, which is also known as nomenclature, Ayodele 2017 defined taxonomy as the study of classification, identification, and nomenclature. These three concepts are therefore the foundation of any taxonomic work: identification, classification, and nomenclature of plants. Identification is the process of determining the name of a plant by comparing its visible characteristics with those of already known and documented species. For example, if a forester comes across a broadleaf tree with milky sap and opposite leaves in a rainforest, they may suspect it belongs to the family Apocynaceae, but by narrowing down the features and using a dichotomous key, the plant may eventually be identified as Ravovia vomitoria. Correct identification is the first step before any meaningful silvicultural intervention, such as propagation, conservation, or reforestation. Classification, on the other hand, is the process of organizing plants into groups, also known as taxa, based on shared characteristics. These groups are arranged in an hierarchical system ranging from broader categories to more specific ones. The main taxonomic ranks are therefore kingdom, division, class, order, family, genus, and species. Plant nomenclature is the scientific system by which plants are assigned names governed by standard rules. In taxonomy, each plant species is assigned a binomial name, that is, the specific and the generic name, following the principles laid down in the International Code of Botanical Nomenclature. This naming system ensures consistency and universality across countries, languages, and scientific disciplines. Therefore, plant names are not abstract, as most people believe. They have meanings. Sometimes plant names are linked to names of people or places where they are first described or discovered. In other cases, plant names may also be tied to attributes of the plant. Formation of plant generic and specific names. The formation of a generic name of plants follow certain linguistic and grammatical conventions rooted in Latin, which has traditionally served as the language of science. When naming genera in honor of individuals, certain guidelines must be followed. If a generic name is to be constructed from the surname of a person for a new plant which has just been discovered or described, the following rules therefore apply. 1. If the person's name ends in a vowel other than A, suffix A is added. For instance, the name Josiu becomes Joshua. 2. If it ends in consonant, add suffix IA. For example, Bohin becomes Bohinia, as you can find in the plant Bohinia monandra. While Vernon turns to Vernonia, as in the case of Vernonia amygdalina, which is your bitter leaf that we usually take. 3. If the name ends in A, the suffix EA is typically added. For instance, Cola becomes Colia. For names ending in ER, adding A transforms the name into a generic one, such as Kenna becoming Kenera. Where a name is ending in US, the US is removed and replaced with A, as seen in Dillenius, which becomes Dillenia. Therefore, to form a specific name, which is the second name of the plant, from the first name of person, the following rules apply. If the name ends in a vowel other than A, suffix I is added. For instance, the name Birui becomes Burui. If it ends in A, e.g. Balancer becomes Balance. If the name ends in consonant, II is added. For example, Bram becomes Branai. Therefore, to form the name of plant from my own name, I want you to cross-check the above rules if Onephilia afredi conforms to the rules. Then what is silviculture? Silviculture is the art and science of managing forests to meet specific goals such as timber production, conservation, and restoration. It deals with how forests are established, grown, and renewed, whether in plantations or natural settings like biosphere reserve, helping to maintain biodiversity and ecosystem health. Nigerian forests are rich in biodiversity, but they are faced with serious threat from deforestation, mostly driven by agriculture and other land use. Silviculture alone cannot therefore solve the problem of deforestation. It must work alongside other fields like taxonomy, ecology, and forest conservation, according to FAO 2025A. Different silvicultural systems and practices have been developed in the tropics to address this issue. These include tropical shelterwood system, Tanger system, selection cutting, natural regeneration, enrichment planting, and so on. My contribution to knowledge. Plant propagation and silvicultural techniques. One of my contributions to silviculture involved developing vegetative propagation methods for two ecologically important indigenous tree species in Nigeria, Pycnanthus angolensis and Xanthoxylon xanthoxyloides. These species face difficulties in natural regeneration due to seed-related constraints. Pycnanthus angolensis, though valuable for timber and combo butter production, has recalcitrant seed that stop poorly. Xanthoxylon xanthoxyloides, widely used in traditional medicine as an insect repellent, is often destroyed on farms because of its prickly nature and limited seed availability. To address these challenges, Onefeli and Akinyele 2012 examined the stem cuttings from 3 nodal positions treated with varying concentration of indobutyric acid. Although no rooting was recorded, significant callousing was observed at higher IBA concentrations, especially at 3,000 ppm. The nodal positions of cotton had no significant impact, but hormone concentration significantly improved callousing, as shown in tables 2 to 4. These findings suggest potential for vegetative propagation of both species using improved hormonal treatment. In another study, Onefeli and Adesoye 2014 investigated the early growth performance of selected indigenous Kaya senegalensis, Kaya grandifoliola, Afcelia africana, and exotic species, Melina arborea and Tectona grandis. Contrary to prevailing assumptions, Kaya grandifoliola and Afcelia africana performed competitively, with Kaya grandifoliola significantly outperforming the exotics in height growth. Table 5, figure 7 to 9. This result suggests that indigenous species under similar conditions can match or even exceed the exotic ones in early growth. The study highlighted the silvicultural potential of native species and supports their inclusion in afforestation programs. Onefeli and Akiele 2014 conducted a pioneering study on macropropagation of Denetia tripetala, which is popularly known as pepper fruit. Using the stem cuttings from mature trees, the study revealed that a combination of 2000 ppm and upper nodal cuttings produced the highest rooting sources of about 95%, while hormone concentration and nodal position significantly affected rooting and survival. They did not significantly influence growth parameters, as shown in table 6, figures 10 to 13. This highlights a cost-effective propagation technique for conserving and cultivating these non-timber forest species. Some other indigenous tree species to which my work with colleagues has contributed significantly in terms of propagation and silvicultural techniques include Ostonia congensis, Clestopholips patens, and Rhizophora resmota, according to Jegede et al. 2020, table 7, 8, and 9. Rhicinodendron hydaloti, Onefeli 2021, table 10 and figure 14. Idegardia bateri, Akinyele et al. 2020, table 11, figure 15. And Rhicinodendron hydaloti, Onefeli et al. 2019, tables 12, 13, and 14. Floristic diversity and ecological assessment of Nigerian forest ecosystem. Sustainable forest begins with knowing what we have, and taxonomy is the foundation for understanding the structure and health of any forest. Before we can manage forests effectively through silvicultural intervention, it is essential to assess the diversity of the existing species and their ecological roles, as well as their impact of human activities on them. My approach to this is what I often describe as a taxonomic ramble, which is a systematic but practical walk through the forest to document what species are present, in what quantity, and in what condition. This taxonomic rambling was carried out by Onefeli 2018 in the Olekwemeji Forest Reserve, and only about 45 individual plant species distributed into different families were identified. Tables 15, 16, and figure 16. Figure 17 shows that the savanna section of the forest was more diverse when compared with the fringing forest. You will agree with me that there is lack of up-to-date information on the flora of Nigeria. This is because most of the available information found in the flora of Nigeria come from studies conducted and documented many years ago. Sadly, many silvicultural studies continue to focus on a few familiar indigenous tree species, mainly because you cannot improve or cultivate what you do not know. Also, most of the existing forest inventories and silvicultural works are focused mainly on trees, often ignoring other plant life forms such as herbs, lianas, and epiphytes, mainly because they are considered to be of lower economic value. However, many of these overlooked plant species are quite valuable. Species like Ancistrocladus coropensis, Afrostyrax lepidophilus, Amistolotype tenuis, Acistrocarpus densispinosus, Anisophilia disticha, Begonia potamophyla, Capilospermum serratum, Cyanastrum cordifolium, Salicodiscus gabonensis, Ensia crenata, Mosua brunonis, just to mention few, are rarely studied in silvicultural research. For instance, Acistrocladus coropensis a liner has been found to contain an alkaloid known as methylamine B, which shows strong in vitro activity against HIV-1 and HIV-2, according to McKay et al. 1996. Because of this gap in knowledge about Nigerians' flora, and as part of my ongoing postdoctoral research, I led a team of researchers to carry out a comprehensive survey of key forest ecosystems in southern Nigeria, specifically Cross River National Park, Afi Mountain, and Imbe Mountain. In the course of the survey, I witnessed mysteries in diversity of plants encountered in the forest, which prompted me to publish what I titled Mystery of Ontogenic Plant Diversity, Onefeli 2024. One of such mysteries is the one I encountered in Sarcestis mirabilis and Sarcestis cameroonensis. In my first 3 weeks of the survey, I was confused with these plants having diverse morphological physiognomies. I unknowingly tagged the same plant species as different unknown species entirely, judging from their striking morphological differences. After some research about the plants, I eventually discovered that the mystery in their morphological variations to be ontogenic. In the case of Sarcestis mirabilis, at juvenile stage, the leaves are entire and silver maculated, figure 18. After a while, the color changes gradually to green, figure 19. At this stage, the plant begins to send its roots to the adjoining trees by both horizontal and vertical anchoring roots, figure 19. For Sarcestis cameroonensis at the juvenile phase, figure 20, they usually creep on the forest floor with oval leaf shape. As they grow older, their leaves begin to perforate longitudinally, mostly climbing on a host tree with their roots anchored on the stems, figures 20 and 21. At the adult phase, they have fully developed roots anchored on a host tree. Another morphological characteristics of an adult Sarcestis cameroonensis is successive rosettes of leaves, usually separated by ascending flagelliform at the narrow part of the stem, figure 21. The adult phase of Sarcestis cameroonensis is also characterized by inflorescence and infructescence, figure 22. In a space of 7 months, we surveyed about 700 plots while camping continuously in the forest, figures 23 to 25. We photographed all accessible plant species and collected about 3,000 virtual specimens. So far, we have identified around 400 plant species, while about 100 species remain unidentified. Some of these virtual specimens are currently undergoing taxonomic verification at the Royal Botanic Gardens, Kew, by colleagues in the Herbarium in United Kingdom. By the end of this project, my team and I hope to publish a new and more comprehensive flora of Nigeria, complete with colored photographs and detailed taxonomic descriptions of the plant species currently existing in Nigerian forests. My research into the ecological health of Nigerian tropical rainforest has included an in-depth assessment of 3 species diversity and above-ground biomass across key rainforest ecosystems. In one of these studies, Onefeli et al. 2021 investigated 7 forest reserves in Nigeria, including Agowu, Akure, Cross River South, Ekuri, Ingrigon, Olua, and Shasha. The finding shows notable ecological variation across the reserves, figure 26 to 28, table 17. The variation across sites demonstrate the need for location-specific silvicultural strategies, morphological taxonomy of plants. Before DNA was used in plant classification, scientists primarily relied on plant morphology, such as leaf shape, flower structure, and stem anatomy. Many taxa were originally described using morphological features, which still remain important, particularly for species lacking molecular data. Onefeli and Oladele Akin in 2020 examined 4 stecular species: Steculia tragacantha, Steculia rhinopetala, Steculia oblonga, and Steculia setigera, using macro and micro morphological features to resolve their taxonomic confusions. Tables 18 to 21 and figures 29. Epidermal characters showed interspecific variation. Trichomes were present in Steculia stetigera and Steculia tragacantha, but absent in others. Stomata types vary among species. Principal component analysis explained about 85% of variation based on stomata and epidermal traits. Table 22. Phylogenetic analysis, figure 30, supported species distinction, with Steculia oblonga appearing as the earliest species evolved. Onefeli and Kinde 2021 used leaf epidermal traits to distinguish 5 medicinal apocynaceae species: Ostonia bunei, Olarena floribunda, Ravuvia vomitoria, Tevetia nerifolia, and Vuacanga africana. Clear interspecific differences were observed. Tables 23 to 25. Figure 31. These studies highlight the relevance of morphological taxonomy in identifying and classifying plant species, especially where molecular tools are not available. Molecular taxonomy. One of my earliest molecular taxonomy studies using ISSR markers focused on Rhinodendron eudelota, a medicinal tree with limited taxonomic clarity due to overlapping morphological traits. Results, tables 29 to 31, figures 33 to 36, showed significant molecular variability in the population of Rhinodendron eudelota in Nigeria and suggested deeper taxonomic reconsideration. Furthering this, Onefeli and Stanis 2019 studied the phylogeny of Combretaceae using the RSBS gene. 3 major groups emerged, with Combretum and Terminalia appearing polyphyletic, figure 37, while Conocarpus, Quisqualis, and Mestemon appeared monophyletic. Close clustering of Quisqualis and Calopixis with Combretum and Teleopsis with Terminalia, figures 38 and 39, supported existing proposals for taxonomic realignment. This confirmed the utility of RBCL in resolving complex taxonomic relationship. Following this, a focused study on Terminalia superba and Terminalia ivorensis, which are known as the white aphara and black aphara, and AFLP markers were employed to resolve difficulties in distinguishing seedlings. Out of 740 fragments generated, 619 were polymorphic, table 32. One primer combination produced 9 unique markers separating both species. Analysis of molecular variance showed that 61% of genetic variation was between species, table 33. Principal component analysis and cluster analysis confirmed 5 individuals initially tagged as Terminalia ivorensis were genetically closer to Terminalia superba, figures 40 to 42. This validated AFLP as a reliable tool for species identification and detecting hybrids. In a related study, Danelia ogia and Danelia oliveri were differentiated using 3 barcode regions, MATK, TRNL, and ITS. Onefeli 2021, a total of 46 sequences from NCBI were analyzed. MATK had the highest discriminating power among the single loci, table 34, figure 43. But the combined MATK, TRNL, and ITS dataset provided the most accurate results, separating both species with over 93% accuracy, figure 44. Although no distinct barcoding gap was found, figure 45, the combined loci effectively resolved the species. Conclusion. This lecture has explored the crucial intersection between taxonomy and silviculture, emphasizing their complementary roles in addressing Nigeria forest management challenges, from morphological to anatomical studies, molecular techniques such as AFLP and DNA barcoding. It is evident that taxonomy research provides the necessary foundation for silvicultural decisions, including species selection, domestication, conservation, and restoration. Through personal academic experiences and original research contributions, including the molecular characterization of Terminalia superba and Terminalia ivorensis, this lecture has shown how taxonomy enables accurate species identification, even at juvenile stages, a major limitation in morphological approaches. The result of these studies also underscore the importance of integrating molecular data into forestry practices to resolve ambiguity, delineate species boundaries, detect hybrids, and conserve genetic resources. Therefore, sustainable forestry in Nigeria cannot be achieved without strong taxonomic backbone. Silvicultural interventions, whether for commercial timber production or ecosystem restoration, must be taxonomically informed. Recommendations: institutionalize taxonomic integration. Forestry Research Institute, faculties, and professional bodies should deliberately incorporate taxonomic training, especially molecular taxonomy, into their silvicultural programs and curricula. Strengthen herbaria and DNA laboratories. National and institutional herbaria should be well equipped and digitized. Likewise, forestry-focused molecular laboratories should be established or upgraded to enable accurate species identification and genetic assessment. Promote applied taxonomic research. Funding bodies and government agencies should support applied taxonomy studies, particularly those that resolve field-level challenges, such as species misidentification in nurseries, afforestation programs, and biodiversity conservation planning. Encourage multidisciplinary collaboration. Stronger collaborations between taxonomists, silviculturists, geneticists, and ecologists should be fostered to deepen the understanding of forest ecosystems and improve sustainable management practices. Acknowledgement. My first and deepest appreciation goes to Almighty Heavenly Father, Elroy, the God who sees me. He has preserved my life and made it possible for me to stand here today and deliver this lecture. May His holy name alone be glorified. Amen. I am profoundly grateful to the Dean of the Faculty of Renewable Natural Resources, Professor Adejoke O. Akinyele, who also served as my MSc and PhD supervisor. She not only gave me this opportunity to deliver the faculty lecture, but has also taken a personal interest in my academic journey. One of our words of encouragement that always keep me going is, Alfred, you cannot have a perfect work. Bring what you have. I also deeply appreciate the head of Department of Forest Production and Products, Professor A.O. Omole, for his unwavering support and motivation. It has been a pleasure working closely with him. I sincerely thank all the academic colleagues in the Department of Forest Production and Products and Social and Environmental Forestry who have contributed in one way or another to my academic career and to this faculty lecture. I specifically acknowledge Professor L. Popola, O.Y. Ogusanwo, A.O. Oluwadare, S.O. Jimon, I.O. Aziz, B. Ajayi, A. Ayeloja, S.O. Olajibe, and Doctors O.F. Falade, D.A. Akitude Alo, P.O. Ige, Oyinlola A. Fashoro, A.O. Adenaya, and Mr. C. Clement Olayanu. My heartfelt gratitude goes to the late Professor O.I. Ajewole, whose contribution to my academic development was truly impactful. I appreciate the consistent support of the non-academic staff in the Department of Forest Production and Products, H.T. Fatoba, and many others, as well as the non-academic staff of the Department of Social and Environmental Forestry. I specifically thank Professor B. Omitoyin and E.K. Ajani for their constant encouragement and support in my academic journey. My gratitude also extends to other members of academic staff of the Department of Aquaculture and Fisheries Management, as well as Wildlife and Ecotourism. Professor I.A. Ayodele, Professor Adetola Jenyoni, Professor A.O. Akinwole, Professor Fumilola Ajani, Professor S.O. Ojo, Professor Abosede O. Omonona, Professor Flora E. Olaifa, and Dr. Sinyambola A. Omitoyin, Dr. Yetunje Agbeja, Dr. A.O. Ajiboye, Dr. A.T. Adeyanju, Dr. O.O. Oyebola, Dr. O.K. Karim, Dr. B. Odunton, Dr. O.M. Koka, Dr. Oluwakemi Akpata, Dr. S.O. Olajesu, Dr. A.T. Adetuga, as well as Mr. F.D.P. Satimeyin and Mrs. Josephine O. Adebayo for their continued encouragement and support. I am especially thankful to Professor J.S.A. Osho for his fatherly advice and constant encouragement to me and my family, so much that my wife fondly called him Godfather. I also thank Professor I.O. Aziz, Professor S.O. Olajibe, and Dr. D.A. Akitude Alo for reviewing this manuscript and offering useful suggestions despite their busy schedules. I have greatly enjoyed working with Dr. Babatunde Ojebui, the warden of the Kutio. In my capacity as an assistant or warden, my appreciation also goes to Dr. Odi Adeniji, my senior and former assistant warden, as well as other staff of Kuti Hall for their collegiality and support. My heartfelt thanks to Professor E.A. Ajaf and Dr. Aderonke Ajaf, leaders of the University of Ibadan Christian Family Fellowship, for their prayers and guidance. I also thank all members of the fellowship for their encouragement. Similarly, I deeply appreciate my pastors at Redeemed Christian Church of God, Peculiar People's Parish, Pastor Bamidele Adineji, the PICP Oyo 18, Pastor Patrick and Yemisi Okesojo, Pastor Kunle Oyegbile, Pastor Adebi, and Pastor Dorcas Oje, for their prayers, teaching, and spiritual leadership. I sincerely thank Assistant Pastor Taiwo Akibiseyin and all members of Peculiar Voices, the choir where I joyfully serve. Your fellowship has been a blessing to me. I also sincerely acknowledge the unwavering support of the non-academic staff members of the Dean's Office, whose contributions have been invaluable throughout this journey. Mr. Tubosun Afolabi, the FO, Mrs. Comfort Alo, Mrs. Olarewaju Nurat Ibijoke, Mrs. Olubumi Obaro, Mrs. Moses Fashina, and Mr. Gbenga Adedeji. Your administrative assistance and commitment to duty are truly appreciated. To my landlord and neighbor, the Honorable Chief Oshati Olatunde Emmanuel, the member Ondo State House of Assembly, Ose constituency, and Dr. Mrs. Oshati, other neighbors, Dr. Monsuru, Mr. Adeyemo, and others. Thank you for your friendliness, constant encouragement, and communal support that have made home life peaceful and pleasant. To my friends and research colleagues, Professor P.O. Adesoye, Professor F.D. Babalola, Dr. A. Adesoji, Dr. T.I. Borokini, Dr. O.P. Agu, Dr. F.N. Ogana, Dr. A. Adaja, Dr. Precious Nguli, Dr. V.N. Olorunimbe, Mr. O.A. Ojebode, Ms. Jesutobi Olorunmola, Mr. A. Nkanu, and Mr. A. Takumbu. Thank you for your friendship, academic collaboration, and support through the years. I remain grateful to Professor J.D. Oluwokudejo, Professor A. Ayodele, Professor A. Jayola, and Drs. L.A. Adebisi and E.O. Shokefun for the strong foundation they laid in the field of plant taxonomy, which has shaped my academic path. Special thanks to Professor S.O. Jimo, Director of SESDEF, who gave me my first opportunity to work as a research assistant during my undergraduate days. That first research exposure and my first salary came through him, and I will always be grateful. I will forever be grateful to the late Professor B.O. Agbeja and Dr. Yetunde E. Agbeja, who warmly accommodated my family and me after secured my appointment at University of Ibadan, without asking for anything for years in return. Their kindness and generosity remain deeply appreciated. I sincerely thank Professor Vidmata Stanis for hosting me in his molecular laboratory and supervising my Erasmus study at Vitatus Magnus University Agricultural Academy in Lithuania. I also appreciate Dr. Bairute Frax, from whom I acquired valuable bioinformatics skills at the Department of Orchard Plant Genetics and Biotechnology, Lithuanian Research Center for Agriculture and Forestry, Bapte, Lithuania. I sincerely appreciate my colleagues and students in diaspora, Dr. Shakrudin, Dr. F.N. Ogana, Dr. A.R. Majekubade, Dr. K. Afolayan, Dr. S.M. Adeyemo, Dr. A.A. Oteshile, Dr. A. Oyelere, Mr. S.O. Shitu, Mrs. Azizat Mayokun Shitu, Ms. Praise Ifedayo, Mr. J. Oluborode, Mr. A.S. Olabamiji, and Mr. T.M. Adeyemo for their generous support towards this faculty lecture. I am also grateful to Mrs. Caroline S. Olori, former conservator of Park, and Becky Ikpeme, deputy conservator of Park, Cross River National Park, for their encouragement and support. I acknowledge all my past and present undergraduate, master's, and PhD students for their dedication and support towards this lecture, whether through fieldwork, lab analysis, academic discussions. Special mention goes to Blessing A. Shaibu, H.T. Fatoba, E.T. Lori, I.O. Yusuf, Oluwapelumi, I. Daniel, O.H. Akinyade, Shalom E. Egbebi, Rukayat O. Ajiboye, Salam, Sama M. Omatara, Oreoluwa A. Adeyinka, Oluwakemi, I. Oluranti, T. A. Babalola, V. O. Jackson, L.P. Kinde, A. Grace Fabowale, Amarachi Love Nwachukwu, C. Emeda Ndwagubam. Emeda was the one who prepared the slide that you are viewing now. Thank you so much. Rabiat Ahmad, Boluwatife P. Olawumi, Jessica D. Salami, Oyindamola O. Oluwaseun, Oluwaseyi O. Emevon, E. Adaraniwon, Shukrat O. Adeniji, Jesu Tosin Victoria Ogunfowokan, Zainab B. Abu Fatai, among many others that I may not mention here. Your inputs have enriched this lecture in many ways. I also appreciate my in-laws, Mr. and Mrs. Iwebenua Patrick, Ikechukwu Iwebenua, Oluwatimiro Mercy, Jacob Iwebenua, Rebecca Iwebenua, James Iwebenua, and Ngozi Iwebenua for their love, prayers, and support. I want to thank my entire family, my late parents, Mr. and Mrs. Francis Onefeli, whose prayers and sacrifices laid the spiritual and educational foundation of my life. I also appreciate my elder brother, Peter Onefeli, whom God used to sponsor my education from undergraduate level. My siblings, Uzonwa Aladejobi, Mary Ogunyakin, and many others have remained pillars of support through their prayers, encouragement, and love. I sincerely appreciate Dr. B.C. Falemara, Mr. Anoluwapo Ezekiel, and all other members of the media team for their dedication and support towards the success of this faculty lecture. To all distinguished colleagues, friends, and well-wishers present at this faculty lecture but not individually mentioned, please accept my heartfelt appreciation. Your presence means a lot to me. Above all, my deepest gratitude goes to my darling wife, Mrs. Oluchukwu Joy Onefeli, whose unwavering support, prayers, patience, and love have been my anchor through the highs and lows of this journey. She is truly my joy. And to our beautiful daughter, Esther Ifoma Onefeli, who brings sunshine to our life daily, I say thank you and I love you dearly. To God alone be all the glory. God bless and thanks for listening.
As we wrap up with the anthems, I would like to quickly acknowledge the presence of the Dean of Faculty of Education, Professor Adegbeson, and the Dean of Faculty of Nursing, Professor Priska Adejumo, and also the Dean of Faculty of Environmental Design and Management, Professor Ekmengbemi. Can we take the anthems this time around?
Cherished
Let's remain standing until the academic procession files out.